Now we're going to be looking at some non-living entities. We're going to be looking at viruses and the structure of viruses and how they multiply. And why did I say they're not living? That's what we're going to find out. So let's look at the position of viruses along the spectrum of all our microbes here. These are really unique entities. They are non-living and they are known to infect every type of cell. They're found abundantly on our planet. They're found in the seawater, and the seawater can house 10 million viruses per milliliter. 1 milliliter is like a cc in a syringe. Let's see, feces can contain up to 100 times as many viruses. Viruses might represent about 270 metric tons of organic matter on Earth. That is a lot. We're just really beginning to understand the impact that viruses have on our environment. We did not understand the transmission of viral infections such as smallpox or polio for many years. Louis Pasteur was heading in the right direction in his studies when he hypothesized that rabies was actually caused by a living thing that he believed was smaller than a bacterium. He later developed the first vaccine for rabies and proposed the term virus to name this group. In the 1890s, we discovered the first information about the unique characteristics of viruses. Avansky and Bijernik showed that the tobacco mosaic virus, which is actually used now in genetic engineering, was causing a disease in tobacco plants. Leffler and Roche then discovered another agricultural virus that caused hand-foot-mouth disease in bovines, which are cattle. These scientists studied infectious fluids, and they found that viruses did not filter out of fluids like bacteria did. We've since found out that viruses have a definite size, shape, and chemical composition, but they are considered acellular, and as I've already said, they are not considered living. So the viral debate. This is like Facebook from the 1800s where everybody's debating. The rather unusual structure and behavior of viruses have led to many debates about viruses and their relationship to the rest of the microbial world. Like, are they living or non-living? Are they inert or active? So two sides here we could talk about. You could argue this point from. Since viruses are not able to multiply independently from the host cell, and what that means is they cannot copy their DNA or RNA on their own. They cannot replicate, which is one of the conditions for considering something living. They are not living things. And so one argument is that they should be called infectious molecules. And so even though viruses do not exhibit most of the life that or the life processes that we find in cells, they do direct the processes in the cells. And so some say that we should say that they are a little bit more inert than just lifeless molecules. But viruses are really better described as active or inactive rather than alive or dead. Recent discoveries have led us to understand that viruses are important in forming cells and other life forms. So when they infect other cells, they change or influence their genetic makeup. We actually have found and believe that about 8% of the human genome, our actual DNA, is viral. And up to 20% of the bacterial genome is viral as well. Viruses are what we call an obligate intracellular virus. That means they are obligated, or they must live within, intra means within, a cell. They have to have another cell in order to survive, so to speak. They cannot replicate themselves unless they're in a host cell. Viruses are very different than their host cells in their size, structure, their behavior, and their physiology. This is a really good summary slide of all the properties of viruses. I'm not going to read through all of these because you can read, and there's no point in me just reading straight from the slide, okay? And so we're going to talk about, or we have talked about lots of these things, but this is a really good summary slide to be sure that you know. So viral nomenclature is a little similar to the nomenclature that we use for bacteria and other microbes. Remember, nomenclature is how we classify and name organisms. So for many years, animal viruses were classified based on what their host was or the disease they caused. We've changed that somewhat, and our newer classification systems will emphasize things. They will include the host and disease that's caused, but they will also refer to the structure, their chemical composition, and sometimes their genetic makeup. Okay, so we have an international committee on the taxonomy of viruses. We've got about 8 orders and 38 families. You don't have to learn all of these orders individually, but you will learn about them and you will learn them just in a little bit different method. It's not like we're going to go through King Philip came over for grape soda like we did with the eukaryota, okay? But we will get into the families and orders of viruses. Okay, so here we have another NCLEX prep question. What statements or which statements are going to be accurate regarding properties of the viruses? And let's select all that apply. Okay, so pause this, read this, and find the answer. And I do pull these from your textbook, so if you've already answered them, you can kind of fast forward through this part. But if not, these are really, really good practice questions. They help you to review both our material and to prepare for the NCLEX style questions. And this also can be similar to questions you may see on other entrance exams. This just happens to be geared towards more the NCLEX, okay? So after all that, hopefully you've picked your answers, and the answers here are going to be D and E. Viruses do not contain enzymes for metabolic processes, which is one of the reasons they're not considered living. And they do contain either DNA or RNA. They can have either one. And remember, sometimes they can convert one to the other. And that's what we call a retrovirus, okay? So this is kind of reminiscent of that size and scale. And they actually have been made, some of the pictures look the same, but I don't know, they may be different, but it almost looks like it's made from the same place. But so our smallest infectious agents, with a few exceptions, are going to be viruses. Okay, so they are way down. Let me put up the little way down here, all right? And this is an E. coli bacterium, which is very, very small, a yeast cell, which is also small. And so you can tell by looking at these. Here's a hemoglobin, which is a protein molecule. It's the protein that carries your oxygen, and then viruses are just a little bit larger than that. And so these are the smallest infectious agents, and of course there are always a few exceptions to pretty much every rule, but they are dwarfed in size by their host cells. Okay, so very much dwarfed in size. Okay. And so there are more than 50 million polioviruses that could fit into an average human cell. I don't know why this is not letting me turn my little thing here off. Okay. Common animal viruses are going to range from the smallest being parvoviruses, which are going to be around 20 nanometers. Nano means 1 times 10 to the negative 9, so that's a decimal and 9 numbers. The largest is going to be around 20. The largest viruses are going to be herpes simplex viruses, around 150 nanometers. And then we also have some cylindrical viruses, which is a little bit longer. You can see this rickettsia is going to be like that. It's usually going to be something like rickettsia. That's actually bacteria. Here's a rabies virus. That's a better example of a somewhat cylindrical, or this Pandora virus here. Okay. So viruses don't really resemble other cells. They're very different. They cannot make proteins. They just don't have the machinery present in order to do that. Their outer surface is composed of repeating subunits. Their organization is fairly simple and compact. They just don't have all the extra materials in order to perform the functions because they hijack the host's cell in order to do those particular things. So they only contain the parts that are needed to invade and control a host cell, plus their DNA or RNA. So these include an external coating, a core with that DNA or RNA, and an occasional enzyme or two sometimes. All viruses will contain a capsid coat. We can't have a naked virus that has only a capsid coat and some nucleic acid. Many of the animal viruses, most of those that affect humans, also have an envelope. Viruses have proteins on their outer surface that allow them to attach to their host cells, and these are called protein spikes. And so you can see here an enveloped virus with some protein spikes, and then this is a naked virus here. All right. And then we also have what we call a virion, and a virion is a fully formed virus that can establish and infect a host cell. Each capsid is going to be made of smaller subunits, smaller little protein subunits, these little individual, they look almost like little beads here. And these are called capsomeres. They're identical. These spontaneously come together and form that capsid coat. We have two different types here for that capsid. One is going to be this helical unit here, and the other is what we call icosahedral, all right? Icosahedral. Enveloped viruses get their name because they take a bit of the cell's membrane with them that forms an envelope. Some of them will butt off the cell membrane, and then others actually leave via the nuclear membrane itself. These guys are going to be more flexible, so they're what we call pleomorphic, pleo, P-L-E-O, M-O-R-P-H-I-C, pleomorphic, which means they actually can have many different shapes. They're not always going to have the same identical shape per every individual little virus. So the simpler helical capsid have rod-shaped capsomeres. They're going to bond together and form kind of like a series of hollow discs. Looks a little bit like a bracelet. During the formation of that, they're going to link to other discs and form that continuous helical structure. And that is where the nucleic acid is going to be coiled. A naked structure, however, they're very rigid, very tightly wound into a cylinder. You can see that in this picture down here. Okay, I don't have this picture down here. A good example of this, and something that is actually used a lot in research today, is the tobacco mosaic virus, which attacks tobacco leaves. We actually use the tobacco mosaic virus in genetic engineering, and if my memory serves correctly, it's as a vehicle to actually get some genetic material into other cells. This is just a good little summary slide of some of those different shapes. I'm not going to just read it word for word to you. You can look at it, but I would definitely be familiar with this. And here are some more. This gets into also the complex capsids, and this is going to be our bacteriophage down here, a T4 bacteriophage. These guys are not going to be symmetrical. They look like little space spots. I'm trying to remember if we've discussed it yet, but a bacteriophage is a virus that actually infects bacteria. The number of viral genes is very small compared to those that a cell has, obviously, because it's less complicated. We can have 4 genes up to hundreds, all right? So they can have only the genes that are needed to evade that host cell and take over their cellular machinery. As I've already said, they have only those. The genome, is actually the sum total of all genetic material that an organism has. Cells contain DNA and RNA, but remember, viruses have only one or the other. So, for example, hepatitis B only has 4 genes, where some herpes viruses have hundreds. Viral nucleic acids can differ in the structure of DNA and RNA. They can have single-stranded DNA, they can have double-stranded RNA, and the normal, of course, double-stranded DNA. They can have linear forms, they can have circular forms. Some RNA has to actually be converted before translation. Some are just pieces of genes, and we have retroviruses, which will convert their RNA to DNA. We've got positive sense and negative sense. Positive sense RNA is that is already ready for translation. Negative sense is going to need to be converted before translation can occur. Okay, and then segmented is just going to be individual genes on little separate pieces of RNA, so they're in segments. Here are some examples of each configuration of nucleic acid. I would go ahead and make flashcards or add these to your existing flashcards if you've listened and started those, because this is the type of information that you are going to need to know at some point. And generally, I expect you to know it in that unit and then to continue with that and retain it. So you're going to want to refresh your mind on these and kind of go through them as they come up again, especially when we get to the end and we learn about what affects certain systems. Okay, so we have double-stranded DNA. We'll find that in the variola virus, which causes smallpox. We can also find that in herpes simplex 2, which causes genital herpes. And I believe I've already said this, but be sure to differentiate and understand the difference between the name of the virus and the disease it causes. Sometimes they're extremely similar, but sometimes they are not, okay? You're also going to see a lot of different herpes viruses, and you're going to need to be able to differentiate those. So find a way to keep them straight. Parvovirus is a good example of a single-stranded DNA. This is the one we're talking about that causes arrhythmia infectiosum, not talking about parvovirus that affects dogs, two different types of parvoviruses there. So as far as RNA viruses, a good example of a positive sense single-stranded is poliovirus, which causes polio or poliomyelitis, all right? Then we can have the single-stranded negative sense in an influenza virus, which is the flu, and you can see the disease and the virus name there are the same. Double-stranded RNA is going to be found in rotavirus, which is a very common gastroenteritis or stomach virus that you see a lot in children in preschool, kindergarten type age. Single-stranded RNA, which is going to include reverse transcriptase, positive strand, is what HIV, human immunodeficiency virus, is going to be, and that is going to cause the disease acquired immune deficiency syndrome, which is, of course, known as AIDS. Viruses may also contain enzymes needed for specific operations that need to occur in the host cell. So they may contain enzymes that are needed for viral replication, like polymerases, okay, that help to make DNA and RNA, or replicases that will copy RNA. HIV will come with reverse transcriptase to convert that RNA to DNA. That's what makes it a retrovirus. Don't confuse this, however, because viruses completely lack the genes for a specific synthesis of metabolic enzymes. They cannot specifically make metabolic enzymes. Some viruses can actually take things from the host cell away with them, like arenavirus, the family of arenaviruses. Arenaviridae would be the name of that family. They take host ribosomes. Retroviruses actually borrow the host tRNA molecules.